Alright, we were discussing the meninges of the brain. Dura matter we have discussed. Now, the next or second layer of dura matter is the next layer of the meninges is the arachnid matter. Look, this is the skull bone, the black line, and then this is the meningeal layer of dura matter. In contact with the meningeal layer of dura matter, this red one is the arachnid matter that always remains and being supported by the thick dura matter. This is the arachnid matter. And the third layer, which I told you, that is this, that covers the brain tissue closely. This green one is the pia mater. I have shown you already the pia mater in the previous lecture. Okay, now what is arachnid matter? The dura matter is a thick fibrous membrane, as you have seen it over here, a thick fibrous membrane, while the arachnid matter, it is a thin, impermeable membrane. It is a thin, impermeable membrane that lines the inner surface of the meningeal layer of dura mater, and the space between the meningeal layer of dura mater and the arachnid matter is very less. Thin film of fluid only lies in this, and this potential space between the meningeal layer of dura mater and this arachnid matter, this is called a subdural space. But you know that the dura and arachnid, they remains in contact with one another. The space between the two is a potential space. That's why the blood vessels or nerves, it pierces the arachnid and dura at one place, because they do not run between the two but simply passes through it at one point, because they are very close to one another. Alright, and then you look, the space between this red arachnid and the green pia, this space is much more, and this is being called a subarachnid space, and this subarachnid space remains filled with the cerebrospinal fluid or CSF. And the blood vessels, it runs over here in this space like this. Veins and arteries, it runs in the subarachnid space, and you can see it over here. Look closely. You can see this is the skull bone, which you removed, and then you can see over here these blood vessels that lies over the pia mater between the dura and arachnid. And this thick membrane that you see over here, this is the dura mater, and this artery which you see over here, this is the middle meningeal artery, the branches of the middle meningeal artery that lies over the dura mater between the skull bone, that is the endosteum or endosteal layer of dura mater, and the fibrous dura mater. And over here now, you look, the dura mater is being again cut. Look, this is the skull bone, and see the dura mater. We cut over here and reflect it. Now this is combined dura and arachnid, because you know the arachnid and dura are in contact with one another. And now you can see these big veins running between the arachnid and pia mater in this space, while when it reaches to the arachnid and dura, it pierces through it. It does not run between these two. While over here, our vessels, it runs between the arachnid and the pia mater, and the space is being filled with the CSF, always remains filled with the CSF, and this, as I told you previously, these membranes and the fluid, it provides a cushion for the brain to prevent it from injury. All right. Now, in some areas, what happens? You look. Let me say, this is the superior sagittal sinus in the dura mater, having the blood, and then deep to it, you can see the arachnid matter. In some areas, the folds of arachnid matter project through the dura mater to enter into the venous sinuses, and it is said that the CSF which lies over here, it passes through these folds and oozes from over here into the sinuses, especially the superior sagittal sinus. And this fold of arachnid matter, this part which is permeable and which enters into the sinuses, especially the superior sagittal sinus, these folds are being called as arachnid villi. Arachnid villi are folds of arachnid matter that pierces through the dura to enter into the venous sinuses, and these folds are permeable, and the CSF oozes from the walls of these villi to enter into the venous sinuses, especially the superior sagittal sinus. And you look over here, you can see this one. Look, this fold from the close, and this fold which you can see over here, this is the arachnid villi, or arachnid also called as arachnid granulations only. Okay, only when the arachnid villi in the young age, usually these villi from the arachnid into the superior sagittal sinus, they remain separate, discrete, and they are being called as arachnid villi. But with the age, what happens? Many arachnid villi unite with one another like this, and it forms such a structure, and now it is being called as arachnid granulations. You look over here, this is the dura mater, and this is the superior sagittal sinus, and you can see this small flower-like structure. These one over here, the small nodules, these are actually the arachnid granulations or arachnid villi. Alright, these arachnid villi or granulations, these are used to put the CSF from the subarachnid space into the venous sinuses, especially superior sagittal sinus. Okay, and now the arachnid matter then covers all around the inner of the skull and the inner of the fibrous layer of the meningeal layer of dura mater, and then this arachnid matter becomes through the foramen magnum continuous with the arachnid matter of the spinal cord. Okay, the dura mater also, the pia mater also, and the arachnid matter also passes from the brain through like this. Over here you can see, this is the pons medulla, and this is the spinal cord. Then the pia mater, this arachnid matter, and this dura mater all become continuous with the meninges of the spinal cord through the foramen magnum. Alright. Now note one point that the arachnid matter do not leave the dura, and the pia matter, it do not leave the brain tissue. Alright, and you can see this pia matter. Look, this membrane, look, it invaginates into the sulci and depressions as well, this white membrane, while the arachnid matter, it lines the skull from the inner side and do not enters into this sulci or depressions, but simply bridges over it. Let me say this is one sulcus. Now the pia mater will go in and then come out, while the arachnid matter will simply pass over it and will bridge this sulcus or this groove. The one is this point. Secondly, at the base of the skull over here, you already know, you look, there are many spaces. The pia mater, it comes over here like this and over here like this, and from this side it enters and then become like this, while the arachnid matter, it simply passes along with the skull bone. And now what you see, what will be the condition? You look over here also, that because the pia is in contact with the brain and the arachnid is in contact with the skull bone or with the meningeal dura mater, that's why at the base of the skull, where there are incongruity between the arachnid and the pia and arachnid, big spaces are being formed over here. And these big spaces, the part of the subarachnid space and area, it becomes very big because of the incongruities between the brain structure. Just like look over here. Look, this is the cerebrum, this is the midbrain, this is the pons, and this is the medulla, this is cerebellum. Okay? Now the arachnid matter, it runs like this, while the pia matter, it comes over here like this and then close to this cerebellum like this and then come out. In this way, over here you can see one of the big space between this pia, this one, while arachnid is over here, space is being formed. In this way, many spaces are being formed between the arachnid and dura mater, and these big spaces between the arachnid and dura mater, they are being called as cisterns. CIS, these are being called as cisterns. Cisterns are what? It are the big spaces between the arachnid and pia mater, especially at the brainstem, where there is incongruity or insimilarity between the arachnid and pia. Because you look, the pia is in contact with the brain tissue like this, while the arachnid is in contact with the skull bone, and this way the spaces are being produced. And these spaces are being called as subarachnid cisterns. Okay. Now one of the cistern, look again over here. Look, this is the midbrain, this is the pons, this is the medulla oblongata, this is the cerebellum. And look, over here is coming the arachnid matter, while the pia mater is in contact with the cerebellum and with the cerebrum. That's why between the pia over here and the arachnid over here, this big space is being produced on the undersurface of the cerebellum and on the dorsal side of the medulla oblongata. This is medulla oblongata frontal side, this is pons medulla, and this is dorsal side. Over here, a big space is being formed, and this big space is called as cerebellomedullary cistern or cisterna magna. You can see over here. Look, the pia mater comes like this, then like this, enters into this space, and then becomes continuous size. While the arachnid mater passes directly like this. In this way, this space, which you can see over here, in this way you can see this is cerebellum, undersurface or inferior surface cerebellum and dorsum of this medulla oblongata. Over here, this space is being called as cisterna magna or cerebellomedullary cistern. It is the space between the arachnid matter and the dura mater on the inferior surface of cerebellum and dorsal of the medulla oblongata. To the boundaries of it, above is the cerebellum and its vermis. Below is the arachnid matter, and on the anterior is the dorsal surface of the medulla oblongata. And you look over here. Look, this cavity, this tent-shaped cavity, this is fourth ventricle. Now over here, at the midpoint of the fourth ventricle, there is one foramen at the midpoint like this. if this is the fourth ventricle, over foramen over here at the midpoint, this is called as median foramina or foramina megendi. Foramina megendi in the foramina megendi in the midpoint of the dorsum or dorsal roof of the fourth ventricle, and the CSF from the fourth ventricle passes through this foramina megendi into this subarachnoid space or cerebellomedullary cistern. So, cerebellomedullary cistern is the space between the arachnid and dura on the undersurface of the cerebellum and the dorsal surface of the medulla oblongata, and on the other side is the arachnid matter, and over here is the fourth ventricle, and median aperture from the fourth ventricle, it put the CSF into this cisterna magna. And you already know the two blood vessels ascend upward. They are the vertebral arteries. These vertebral arteries ascend one on each side of the spinal cord and medulla oblongata, and these two vertebral arteries and its branches for the upper posterior and inferior cerebellar artery. These are the contents of this cisterna magna. The contents of over two arteries, cisterna magna. Okay, these are the contents of okay. Then again look over here, please. Look again. This is this cerebellum. This is the corpus callosum. This you can see. This is the midbrain. This is the pons, and this is the medulla oblongata, and this is cerebellum. This is dorsal surface, and this is ventral surface. On the ventral surface of the pons, over here lies one another cistern at the ventral surface of the pons, which is being called as cisterna pontis. Cisterna pontis lies on the ventral surface of the pons, and over here on this side is the arachnid matter, and over here is the pons, and at the surface of the pons is the pia mater. Look, and this is the cisterna pontis, or pontine cistern, which lies anterior to the over here. You can see in this area. Look, the pia mater comes like this, and then like this, and remains in contact with the pons and descend, while the arachnid matter comes like this. In this way, the space which is being formed over here between the pia mater on the surface of the pons and the arachnid matter, that is being called as cisterna pontis. And you look, the cisterna pontis, you can see, contains all these nerves: 5th, 6th, 7th, 8th, 9th, 10th, 11th, and 12th. These all nerves are being contained in this cisterna pontis, and also over here is the basilar artery and its pontine branches which are being given by the artery to the pons. They are the contents of the cisterna pontis. The third cisterna is being formed over here on the undersurface of this part of the diencephalon, and on one side is one temporal lobe, another side is another temporal lobe, and over here is the pia mater, and over here is the arachnid mater. In this way, the cisterna which is being formed over here, this is being called as cisterna interpeduncularis or interpeduncular cisterna. Look, you look over here, please. This is one crus cerebri. This is another crus cerebri. Look, one crus. This is ventral surface of pons and ventral surface of medulla. This is dorsal surface of pons, and over here you can see over here ventricle, and this midline aperture over here into the fourth ventricle. The foramen over here, it gives CSF to the cisterna magna, while this lateral aperture over here, one over here and one over here, aperture in the lateral part, this is the fourth ventricle. Through this and this lateral foramina in the fourth ventricle, it is being also called as foramina luschka. From it, the CSF passes over here into the cisterna pontis. Okay, and then the third cistern is the cisterna interpeduncularis or interpeduncular cisterna. This cisterna lies between this one cerebral peduncle and this another cerebral peduncle, and superiorly our roof is being formed by the floor of the third ventricle and the mammillary body. You can see over here these two small nodules, these are mammillary bodies, and then this is the infundibulum, and this is the floor of the third ventricle. It make the roof of the interpeduncular cistern over here, and this cistern, then you look over here, it is interpeduncular cistern. This one you can see, this is crus midbrain, and that's the other side, and you can see over here, this is one crus, this is another crus, and then the arachnid matter, they stretch from one temporal lobe to another lobe. They stretch over here. This forms the floor of this cisterna. It forms the floor of this cisterna. Okay. Now this cistern over here, interpeduncular cistern, the boundaries inferiorly is the arachnid matter which stretches from one temporal lobe to another lobe, and on each side is the crus cerebri which I have shown you, and superiorly is the mammillary body and the infundibulum, which is the floor of the third ventricle, and over here lies, you can see, this is the third cranial nerve, this is the third cranial nerve, oculomotor nerve, and also the fourth cranial nerve, and over here the part of circle of Willis, arterial circle of Willis, is also being contained over here. Alright, and then the fourth cistern are being called as cisterna chiasmatica. Look, this is one optic lobe, this is one optic nerve, this is another optic nerve. The two make a chiasma over here. This is being called as optic chiasma. Yes, it is. Look, this is one optic nerve, this is another optic nerve, and the two unite with one another, and this is called as cisterna chiasma. And just in front of it over here lies the cisterna chiasma, that in front of the optic nerve chiasma, and over here the anterior part of the circle of Willis is being present. And in addition to that, some people consider some small cisterna. For example, one cisterna may be present over here, in this sulcus, lateral sulcus, over here, because the pia matter dips into it while the arachnid matter comes. This is called as this cistern of the lateral sulcus. Then somewhere inside one vein, and that is the cistern of great cerebral vein. Some people consider it. Look over here again. This is the cerebellum. This is cisterna magna. This is cisterna pontis, and over here is the cisterna interpeduncularis, and the cisterna chiasma is more than over here. Some people consider it the over here cisterna of great cerebral vein. This cerebral vein, which is not proper, but lower down, you know, over here, this is the cervical spine, this is thoracic spine, and then this is the lumbar spine, and the meninges, they finish at the L2 level of the spinal cord. Then lower down there is no meninges, and this lower space over here, below that is being called as cisterna lumbar or lumbar spine, and we can take advantage that we put a needle over here, and we can take the CSF from over here. All right. This is about the arachnid matter. And now the third or innermost layer, this green one, this is the pia mater. The pia mater is a thin membrane that closely cover the nervous tissue and enter into all sulci and gyri and never leave the nervous tissue, remains with it, while the arachnid and dura mater do not enter into sulci and gyri. It is a thin membrane and it lies and it closely covers the brain tissue, as I have shown you over here, this one, and it enters into the sulci and gyri and never leave it. It is a very thin membrane, and you can see over here, I have shown you again and again that it can be stripped from the brain tissue very easily. It can be stripped easily. It is vascular membrane. It is made up of thin vascular tissue. Look, you can see this thin vascular tissue, and it can be stripped from the brain tissue. And the point is, because it lies everywhere of the brain tissue, that's why it is sulci and gyri as well. While look, one point, that let me say this is the thin pia mater, and this is the and see the arachnid matter. The blood vessels lie over here, and over here is the brain tissue. Now any blood vessel, now if want to go to the brain tissue, what does it do? It takes a fold of pia mater along with it, and then the blood vessels comes over here, and it is being surrounded by this space which is being called as perivascular space, and it contains, you know, the CSF, and then it pierces then the pia mater and enters into the brain tissue. While at another places, what happens? Look, you see the pia mater, fold of pia mater, especially where there is any ventricle of the brain. What happens? That fold of pia mater along with the tuft of capillaries, that is many capillaries, and the surface is being lined by secretory epithelium cells. This fold of pia mater along with tuft of capillaries and this secretory epithelium combinedly, it is being called as choroidplexus. And this choroidplexus is being responsible for the formation of CSF, cerebrospinal fluid. The capillaries over here, it pushes the blood. The fold of pia mater, what does it do? It allows some of the contents to enter the brain tissue and does not allow some. While the secretion is being then added by the secretory epithelium, and combinedly a fluid is being formed which is somewhat different from that of the blood, and it is being called as cerebrospinal fluid. Cerebrospinal fluid is being formed by choroidplexus, and choroidplexus is nothing but it is a fold of pia mater along with tuft of capillaries and epithelium. And this is choroidplexus. Now this is all about the pia mater and the meninges. And now the next topic which we will do, that is the venous sinuses of the dura mater, and we will do it inshallah in the next lecture. Thank you very much. This, if I think you study it properly, study it today. If any difficulty, you please ask me many questions, any queries or any difficulty, I will help you to explain. Thank you very much.